Hello everyone, welcome to BioOcean and this is Sheethal with you, and today we are here with our new video and the topic is about virus. And as you all know, this is the currently trending topic as the pandemic is going on in a very increased manner. So I am preparing this video so that you can understand the whole concept of what a virus is, and here in this video we will be discussing about all the aspect of virus like general properties, chemical properties, their morphology, and etc. So I hope that this video will be very much helpful for those who are pursuing in biology. And let's get into the video. Before that, if anyone of you are new to my channel, please go and subscribe to my channel and press the bell icon so that you can get the notifications whenever I upload a video. First of all, we just need to understand about what actually a virus is. So let us define virus. We can define simply them as a kind of infectious agent, right? It causes different type of diseases and like that. So it is merely an infectious agent, but there are so many other properties too. So let's discuss about that. A virus can be described as a small infectious agent that can replicate only inside the living cells of an organism. And viruses can infect all types of organisms from animals and plants to bacteria and archaea. And it is characterized by a single type of DNA or RNA. It reproduces within the living cells and have contributed greatly to molecular biology, genetic engineering, and are very significant in causing different types of diseases. Now let us understand what the general properties of viruses are. Unicellular microorganisms may be classified in descending order of complexity as eukaryotes such as protozoa and fungi, and prokaryotes such as bacteria, mycoplasmas, rickettsia, and chlamydia. Viruses do not fall strictly into the category of unicellular organisms as they do not possess cellular organizations. Even the simplest of microorganisms are cells enclosed within a cell wall containing both type of nucleic acids, that is DNA and RNA, synthesizing their own macromolecular constituents and multiplying by binary fission. Viruses, on the other hand, do not have a cellular organization and contain only one type of nucleic acid, either DNA or RNA, but never the both. And they are obligate intracellular parasites which lack the enzymes necessary for protein and nucleic acid synthesis and are dependent for replication on the synthetic machinery of host cells. They multiply by the complex process and not by binary fission. They are unaffected by antibacterial antibiotics. And they are generally considered as microorganisms in medical microbiology. Viruses occupy the twilight zone that separates the living from the non-living. The demonstration by Stanley in 1935 states that the virus could be crystallized like chemicals in the extraction by Geiger and Scram in 1956 of infectious nucleic acid from a virus that could infect host cells and yet complete virus progeny made it appear that viruses were only living chemicals. Recent advances in molecular biology seem to make the distinction between life and non-life little more than a semantic exercise. As the smallest living unit, viruses offer the best models for understanding the chemistry of life. And the medical importance of viruses lies in their ability to cause a large number of human diseases. Viral diseases range from minor ailments such as common cold to terrifying diseases such as rabies or AIDS, and they may be sporadic like mumps, endemic like infectious hepatitis, epidemic like dengue fever, or pandemic like influenza and the current going COVID-19. They may be localized to circumscribed areas or worldwide, and the control of bacterial infection with antibiotics has enhanced the role of viral infections in human disease. Viruses can cause cancer in animals and birds as well as in humans. Now let us understand the morphology. Starting off with the size, the extracellular infectious virus particle is called the virion, and the viruses are much smaller than bacteria, and it was their small size and filterability. Filterability means the ability to pass through filters that can hold back bacteria that led to their recognition as a separate class of infectious agents, and hence they were known as filterable viruses. As they were too small to be seen under the light microscope, they were called as ultramicroscopic. Some of the larger viruses, such as pox viruses, can be seen under the light microscope when suitably stained. And the virus particles seen in this manner are known as elementary bodies, and the viruses may vary widely in size. The largest among them, such as pox viruses measuring about 300 nanometer, are as large as smallest bacteria mycoplasma. The smallest virus is called parvovirus, measuring about 20 nanometer, nearly as small as the largest protein molecule such as hemocyanin. And the earliest method of estimating the size of the virus particle was by passing them through collidian membrane filters of graded porosity. And the average pore diameter of the filter that permitted passage of the virion gave the estimate size. With the development of an ultracentrifuge, a second method was become available. From the rate of sedimentation of viruses in ultracentrifuge, the particle size could be calculated using the Stokes law. The third and the most direct method of measuring virus size is electron microscopy. Purified preparations of virions may be examined under the electron microscope, either unstained or stained. And by this method, both the shape and size of the virion can be studied. Now let us understand the structure and shape of the virus. The virion consists essentially of a nucleic acid surrounded by a protein called the capsid. The capsid with the enclosed nucleic acid is known as the nucleocapsid, and the function of the capsid is to protect the nucleic acid from inactivation by nucleases and other deleterious agents in the environment. The capsid is composed of large number of capsomeres, which forms its morphological units. The chemical units of the capsid are polypeptide molecules which are arranged symmetrically to form an impenetrable shell around the nucleic acid core. One of the major functions of the capsid is to introduce viral genome into the host cell by absorbing readily to the cell surfaces. Two kinds of symmetry are encountered and in the capsid, namely icosahedral, which means cubical, and helical. An icosahedron is a polygon with 12 vertices or corners and 2 facets or sides. Each facet is in the shape of an equilateral triangle, and 2 types of capsomeres constitute the icosahedral capsid. They are the pentagonal capsomeres at the vertices, which is called as pentones, and the hexagonal capsomeres making up the facets called hexons. There are always 12 pentones, but the number of hexons varies with the group of virus. And in the nucleocapsids with helical symmetry, the capsomeres and the nucleic acid are wound together to form a helical or spiral tube. The tube may be rigid as in the tobacco mosaic virus, but in the case of animal viruses, the tubular nucleocapsid is pliable and may be collided on itself. Not all viruses show the typical icosahedral or helical symmetry. Some, like the pox viruses, exhibit a complex symmetry too. And here, the viruses, otherwise the virions, may be enveloped or non-enveloped. Non-enveloped can be either said as naked. The envelope or the outer covering of viruses is derived from the host cell membrane when the progeny virus is released by budding. The envelope is lipoprotein in nature, and the lipid is largely of host cell origin, while the protein is virus-coded. Protein subunits may be seen as projecting spikes on the surface of the envelope. These structures are called as peplomeres. Peplome means envelope. A virus may have more than one type of peplomer. The influenza virus carries 2 kinds of peplomeres, the hemagglutinin, which is a triangular spike, and the neuraminidase, which is a mushroom-shaped structure. Envelopes confer chemical, antigenic, and biological properties on viruses. Enveloped viruses are susceptible to the action of lipid solvents like either chloroform and bile salts, and specific neutralization of virus infectivity depends on antibodies to the surface antigens. Biological properties such as attachment to the host cell surface or hemagglutination upon the envelope. Some viruses possess additional structural features too, like example, fibrils protrude from the vertices of an adenovirus particle. The shape of the virus particle varies in different groups of viruses. Most animal viruses are roughly spherical, some are irregular and pleomorphic. The rabies virus is bullet-shaped, Ebola virus is a filamentous, and pox viruses are brick-shaped. The tobacco mosaic virus is road-shaped, while bacterial viruses have a complex morphology. That was all about morphology. Now let us understand the chemical properties. Viruses contain, as we earlier said, that only one type of nucleic acid is contained in viruses, like either single or double-stranded DNA or RNA. In this respect, viruses are unique, for nowhere else in nature is genetic information solely carried by RNA. Viral nucleic acids may be extracted by treatment with detergents or phenol, and in the case of some viruses like papovavirus, the extracted nucleic acid is capable of initiating infection when introduced into the host cells. Viruses also contain protein which makes up the capsid. Viral proteins, beside protecting the nucleic acid, also determines the antigenic specificity of the virus. Enveloped viruses contain lipids derived from the host cell membrane, and some viruses also contain small amount of carbohydrate. Most viruses do not possess any enzymes for the synthesis of viral polymerase or reverse transcriptase, which can transcribe RNA into DNA. And now let us talk about the resistance, the viral resistance. With a few exceptions, viruses are very heat liable. There are individual variations, but in general, they are inactivated within seconds at 56 units, within minutes at 37 units, and within days at 4 units. They are stable at low temperature. For long-term storage, they are kept frozen at 70 units. A better method for prolonged storage is lyophilization or freeze drying, which means drying the frozen virus under vacuum. Lyophilized virus can be stored for years and reconstituted when required by adding of water. Some viruses, such as poliovirus, do not stand freeze drying, and viruses vary greatly in their resistance to acidity. For example, we can say that enteroviruses are very resistant to acid pH, while rhinoviruses are very susceptible. And among all viruses are disrupted under alkaline conditions. Viruses are inactivated by sunlight, UV rays, and ionizing radiations, and they are in general more resistant than bacteria to chemical disinfectants. Probably because they lack enzymes, phenolic disinfectants are only weakly viridicial. And bacteria are killed in 50% glycerol, saline, but this acts as a preservative for many viruses. And molar concentrations of certain salt like MgCl2 and Na2SO4 are also protective for some viruses like poliovirus against heat inactivation. And the most active antiviral disinfectant are oxidizing agents such as hydrogen peroxide, potassium permanganate, and hypochlorites. Organic iron compounds are actively viridicial. And chlorination of drinking water kills most viruses, but its efficacy is greatly influenced by the presence of organic matter. Some viruses like hepatitis virus, poliovirus, etc. are relatively resistant to chlorination. Formaldehyde and beta-propiolactin are actively viridicial and are commonly employed for the preparation of killed viral vaccines. And the action of lipid for the preparation of killed viral vaccines, such as the solvents like ether, chloroform, and bile salts, is selective. The enveloped viruses being sensitive and the naked virus is resistant to them. The selective action is useful in the identification and classification of viruses. Antibiotics active against bacteria are completely ineffective against viruses. And this property is made use of in eliminating bacteria from clinical specimens by antibiotic treatment before virus isolations. And this is how the resistance, otherwise the viral resistance, look up. And there are several different types of viruses. We will make a video on that. But one of the virus that infecting the bacteria is called as bacteriophages, and that's like a different aspect of viruses. So if you want to know more about bacteriophages, you can just click on the I button. I've already created a video, and you can access it there. And along with these together, like general properties, morphology, chemical properties, and their resistance, altogether constitute the overall concept of what a virus is, how they work, how do they perform their functions, and what all are their variations, how do they have some kind of similarities, and like that. I hope you got a clear vision of what a virus is, and this video will be very much helpful for you people. If you do so, if you found this video more effective, please do like, share, and subscribe. And also, if you have any kind of doubts or any other suggestions like that, please do comment. Otherwise, you can mail it off to me, and do share this video with everyone who is interested to learn biology. And thank you for watching. That's all for today. Let's see in our next video with a different topic. And thank you for watching. God bless. Thank you.